part of being successful in middle school science is being able to correctly and safely perform an experiment. And in order to perform an experiment, we need to know the scientific method. We need to know the steps involved in performing that experiment. So the steps in the scientific method. This PowerPoint will go through and outline all of the steps to the scientific method, and then we've got an example towards the end of it. So the scientific method involves a series of steps that are used to investigate a natural occurrence. So something that occurs in nature, it's a way for us to test and investigate what's happening. And we'll look at these steps more closely along with terminology that's associated with the scientific method. And my goal is for you to understand specifically how to go about writing a proper scientific procedure as you're performing an experiment. And we'll get some practice in class performing experiments over the course of the year. So the steps in the scientific method, in order, are the problem or the question, observation and research. You're then going to formulate a hypothesis. After you formulate a hypothesis, you are going to make sure that you list all of the materials that are involved in your experiment. You're going to create a procedures list or your experiment list. So it's going to be step by step kind of the recipe for how you're going to go about performing your experiment. You're going to collect and analyze data or analyze results during your experiment. You'll then conclude your experiment. So you'll, after looking at and analyzing all of those results, you'll come to some form of conclusion. And then the last step to the scientific method is communicating your results or repeating your trials over and over again. It's very important that when we communicate results that our experiment must be testable by other people. It has to be able to be performed exactly the way that you perform the experiment. And part of communicating results is allowing somebody else to have the opportunity to recreate your experiment. So the first step, the problem or the question, we're going to develop a question or problem that can be solved through experimentation. So we have to have some form of steps that are involved. We have to work through a series of steps in order to solve that problem. Step 2, observation research. We're going to make some observations, possibly going to do a little background research over the topic that we're planning on experimenting on. That background research can include using web information. It can include going to professional journals. It can include even just observations that you observe in nature. Maybe you come up with a question that pertains to why certain plants grow faster than other ones. In that case, you're making some type of observation of a natural occurrence, and then you might test that that idea that you have, or test that observation that you've made. So the next step, think about it real quick. Do you remember the next step to the scientific method? Step number 3, formulate a hypothesis. Predict a possible answer to the problem or question. So it's an educated guess. It's you coming up with an idea as to what the answer to your question or your problem is. So for example, if soil temperatures rise, then plants will increase. Plant growth will increase. Notice that our hypothesis is written in an if-then statement. If one thing happens, then something else will happen. If we increase soil temperatures, then the plant growth will increase. It is important to write all hypotheses in an if-then statement. Step 4, the experiment. In this step, we're going to develop and follow a procedure. So it's important to kind of think about this area or this step as the step where you kind of make like a recipe. If you were going to be cooking a pie, you want to follow a recipe exactly the way that it's written down. If you miss a step, then your pie might not end up right. In this case, anytime we're creating an experiment, we want to develop and follow procedures that are easily identifiable, they're easy to read, and it's something that somebody else could recreate your experiment through going and following your directions. In this section, we also need to include a detailed materials list. So that includes any materials that you would need for the experiment, also any lab equipment that you might need. So if you were talking about an amount of water that you needed to add to your experiment, that amount of water you then would probably have to write in the materials list that certain amount of water, but you'd also have to write whatever the measuring device is that you're using for that portion of the experiment. So if you're using a graduated cylinder to measure the amount of water, you would put the amount of water in the materials list. You would also put the graduated cylinder in the materials list. And it's important to remember that in all experiments, the outcome must be measurable or quantifiable. Thinking about the root words of quantifiable, the root word is quantity. So if we're talking about quantifiable results or measurable results, we should have a measurable amount. There should be a measurable a quantity that we get in every single experiment that we go through. Step 5, we're going to collect and analyze results. And then in this case, we may need to modify the procedure and come back and retest again. So it's important for us to confirm results that we get by retesting. So we might go back into the procedure after we've collected and analyzed results based on some data that we have found, and we may modify those procedures, and then we want to go back through and we want to confirm the results by testing more than one time. Because on occasion we can get what are called outliers. Outliers are kind of just odd things that happen in an experiment that aren't always the way that your experiment would the outcome would happen. So confirm the results by retesting over and over again. And also in the collect and analyze results section, we're going to include tables, graphs, photographs, maybe diagrams, anything that would make the experiment much easier to visualize and much easier to understand how to repeat it. Tables and graphs are important for quantifying data in the experiment. So tables and graphs can be used to take any results that we get, and then we can look at those tables and graphs and easily understand what the data was that we got. Step 6, the conclusion. So we're going to include a statement that accepts or rejects the hypothesis. So in conclusion, you would say, I accept my hypothesis because, and you would explain why, or maybe you reject your hypothesis. In that case, you would say, in conclusion, I reject my hypothesis, and then you would state why you feel like that is a hypothesis that needs to be rejected. It's important to realize it's okay to reject a hypothesis. It's okay that your hypothesis was wrong. There is nothing wrong at all with a hypothesis being incorrect. You just make sure that in the conclusion you state that. Also in the conclusion, make recommendations for further study or possible improvements to your procedure. It may be something additional that you didn't really test, but you think about it later when you're concluding and you go, well, maybe if I would have done something slightly different, and you come up with an idea of how you might have made that experiment slightly different, and then you would explain those recommendations for further study or possible improvements in that section. And then finally, communicate the results. Be prepared to present the project to an audience. It is very important that all experiments are explained to an audience. Expect questions from the audience. any experiment that is successful, or even not successful for that matter, it helps us to gain knowledge through presenting. So if we're presenting our experiment, even though maybe we rejected our hypothesis, it's still going to allow others to learn from your experiment. Maybe you accepted your hypothesis. And accepting your hypothesis, others can learn from your experiment and how you went about performing your experiment in order to accept that hypothesis. So all of the steps, all 7 steps, can you name them all now? Problem, question, step 1. Observation and research, step 2. Step 3, formulate a hypothesis, remember in an if-then statement. Step 4, an experiment. Step 5, collect and analyze results. Step 6, the conclusion. Step 7, communicate the results. Alright, so let's look at a realistic example that includes some of the terms that you'll need to know when you're actually going about performing an experiment. So here's an example. Formulating a problem or a question. So John watches his grandmother bake bread. He asks his grandmother what makes the bread rise. And she explains that yeast releases gas, and that gas is trapped inside of the bread. The gas is produced because the yeast is feeding on sugar, and so as the yeast releases that gas from feeding on sugar, the bread is going to expand. The dough is going to expand. So John comes up with a problem or question, and he wonders if the amount of sugar used in the recipe will affect the size of the bread loaf. So a word of caution, be careful how you use the terms effect and affect. Effect is usually a noun, and affect is a verb. The effect of sugar amounts on the rising of bread, and then how does the affect, does the sugar affect the rising of the bread? So observation and research. John then researches the areas of baking and fermentation, and he tries to come up with a way to test his question. He wants to know, how can I go about testing that question? So he keeps all of his information in some type of journal. In this case, we've got our binders, and we are going to make sure that we keep all of our information either in some type of science notebook or inside a bunch of paper that is stored inside of our binders. John then takes his, talks with his teacher, and he gives him an experimental design diagram, and this experimental design diagram will help him set up his investigation. The experimental design diagram can be found on our class website. It's basically just an additional sheet that will help you set up your experiment. You'll also be receiving an experimental design diagram for any experiment that you do in science class from me. And your experimental design diagram looks a little bit like this. So it's basically in sentence form with some blanks that allow you to correctly write out your independent and dependent variables, correctly write an if-then hypothesis. You'll identify a number of constants. You also make sure that you pay attention to the number of trials. You'll always have a control as far as the number of trials go. And then we will talk a little bit in there about, or you'll write down in there about dependent variables and how we're going to measure dependent variables. So, formulating a hypothesis. After talking with his teacher and conducting further research, he comes up with a hypothesis, using an if-then statement, of course. And he comes up, if more sugar is added, then the bread will rise higher. So a hypothesis, again, is an educated guess. And it's an educated guess about the relationship between the independent and dependent variables. Okay, Okay, so those variables will be defined in the next few slides. It's important for you to know the difference between an independent and dependent variable. So do you know the difference between an independent and dependent variable already? Think about it for a second and see if you can come up with the difference. An independent variable. The independent variable, or the manipulated variable, it's the factor that is intentionally varied by the experimenter. So it's the one thing that you change in an experiment. So in this case, if John is saying, if I add more sugar to the bread dough, then the bread will rise higher, the one thing that he's changing in each one of his trials is the amount of sugar. So in this case, he's going to use 25 grams, 50 grams, 100 grams, 250 grams, and 500 grams of sugar in his experiment. The dependent variable, or the responding variable, is the factor that may change as a result of the changes made in the independent variable. So thinking back to John's experiment again, he's going to change the amount of sugar, which is the independent variable, and he's expecting the outcome to be that the bread will rise higher. If he adds more sugar, the bread will rise higher. So the dependent variable, or the responding variable, the factor that may change, is the size of the loaf of bread. So in John's experiment, his teacher helps him come up with a list of procedures and a list of materials, and he would write those out in recipe form again. It might be step by step. He might use numbers for each step, and he might use numbers as well for all of the materials. But he's going to make sure that he has an experimental design that is set up so that it is easily testable by somebody else who has not performed his experiment. They can easily repeat exactly what he's done in his experiment based on his directions. Okay, and then his teacher also discusses how to determine the control group. And in this case, the control group is going to be the group that serves as the standard of comparison. So it's the one thing we're going to compare all of the other loaves of bread to. So the control group may be no treatment. So in this case, maybe he makes a loaf of bread without any sugar whatsoever. Or an experimenter selected group. So he might just simply say, my control group is going to be the way my grandma always makes bread. And I'm going to use her recipe as the control. And then I'm going to change the amount of sugar based on whatever her control is. Maybe I'm going to add less sugar from what her recipe is. Maybe I'm going to add more sugar from what her recipe is. So the control group is exposed to the same conditions as the experimental group, except for the variable being tested. So the one thing being changed, and then again in this experiment, the one thing being changed is sugar, the amount of sugar. Everything else must remain the same. Okay, those are called constants. So all experiments should have a control group. So because his grandmother always used 50 grams of sugar in her recipe, John is going to use that amount for his control group. See, he's going to say that the control group for my experiment is baking a loaf of bread that has 50 grams of sugar added to it. John's teacher then reminds him to keep all other factors the same, so that any observed changes that occur in the bread come only from the changes that he has created with the amount of sugar. So the constants in an experiment are all the factors that the experimenter attempts to keep the same. So can you think of some constants for this experiment? There are numerous constants for this experiment. Take a couple of seconds, think about some of the things that are going to stay the same in this experiment, the things that are constant. Constants may include other ingredients to the bread recipe, the oven being used, the rise time, the brand of ingredients, the cooking time, the type of pan used, air temperature and humidity, where the bread was rising, oven temperature, age of the yeast, all of these things may play into how well the bread rises. So all of these things must stay the same for the experiment. So John writes out his procedure for his experiment along with a materials list in his journal. He has both of these checked by his teacher, where she checks for any type of safety concerns. Please be aware, anytime you create an experiment in my class, I will always check your procedure to make sure that there is not any type of safety concern prior to you beginning the experiment. It's important to always wear appropriate safety materials. Anytime we perform an experiment inside of my class, you will have to wear safety goggles. On occasion, you may have to wear gloves. You may have to use a hot pad. You may have to wear aprons. Ladies or gentlemen with long hair, you need to make sure that your hair is always in some type of restraining device, even if it's just a rubber band. Hair needs to be back and out of your eyes. Trials. Trials refer to the replicated groups that are exposed to the same conditions in an experiment. So the number of times you do the experiment over and over again. It's important to have repeated trials, because repeated trials are going to help you be sure that there isn't just some fluke accident that occurs with your experiment. So in this case, John is going to test each sugar variable 3 times. So each amount of sugar he's going to test 3 times. So he'll have 3 repeated trials. So he'll have 3 loaves of bread that have the same amount of sugar added to them. And then he'll do a different amount of sugar, and he'll create 3 loaves of bread for that different amount of sugar. And then he'll probably average the 3 out. Collecting and analyzing results. John comes up with a table that he can use to record his data. And he gets all of his materials together and carries out his experiment. So when John performs his experiment, these are the results that he gets. He measures the loaves of bread by taking the length times the width times the height, and he measures them all in centimeters, so this gives him centimeters cubed. You can see where he's got his multiple trials. He's got trial number 1, trial number 2, trial number 3. You can see where each one of his changes to his independent variable are put inside of the table. So he has 25 grams of sugar added to his 3 trials. He then makes another batch of bread with 50 grams of sugar, and he makes 3 loaves of bread. 100 grams of sugar, and he makes 3 loaves of bread. 250 grams of sugar, 3 loaves of bread. 500 grams of sugar, and 3 loaves of bread. And then he averages them out. So you can see where 25 grams of sugar gave him the average measurement of 758 centimeters cubed. The control group gave him 1,260 centimeters cubed, or I'm sorry, centimeters cubed. And then 100 grams of sugar gave him 1,116, and so on. So in this case, looking at his data, I would notice the largest loaf of bread was from the control group. And the more sugar he added, progressively those bread loaves got smaller. So John examines his data, and he analyzes those results. And he notices that his control worked the best in his experiment, but not significantly better than 100 grams of sugar. So John rejects his hypothesis, But he decides to retest using sugar amounts between 50 grams and 100 grams. So it's important to notice that he rejects his hypothesis in this conclusion. But he wants to know, he wants to be more specific. He wants to know, is there something between 50 grams and 100 grams that gives the largest loaf of bread? So once again, he gathers all of his materials, he goes back to the experiment, and he carries out his experiment again. And here is what he has for results. Can you tell which group did the best? So 50 grams is a control group, and he goes up to 90 grams. Which group did the best? His largest average size is 1612 centimeters cubed. So he finds that 70 grams of sugar produces the largest loaf. His hypothesis then is accepted. He then communicates his results. He tells his grandmother about his findings, and he prepares to present his project in science class. Probably his grandma would like to know, just for the simple fact that anytime she makes bread, she may want to change the amount of sugar that she adds to it. Alright, so observe your world and come up with a question to answer using the scientific method.